Hello everyone. So today our topic is plant nutrition. So let's start. So the first topic in the chapter plant nutrition is photosynthesis. So as we all know, photosynthesis is a process that is carried out by organisms containing chlorophyll. During photosynthesis, plants convert light energy into chemical energy. So as you know, plants are mostly green in color. So the green color indicates that the plant contains chlorophyll, which is contained in chloroplast, as we learned earlier in the chapter of cells. So the chloroplast contains chlorophyll. The chlorophyll traps light energy and converts light energy into chemical energy for the plant. So as you can see here, the reactants are carbon dioxide and water, which react together to form glucose and oxygen. The reaction of carbon dioxide and water takes place in the presence of light and chlorophyll. As you can see here, there is light coming from the sun to the plant, which is uh, trapped by a chlorophyll present in the chloroplast. That light and then carbon dioxide is absorbed by the plant, after which the reaction of photosynthesis take place. So photo is a Greek word for light and synthesis is a Greek word for process. So it is a light process it's a process which uses light, as we know. So plants make glucose. Glucose is the food of plant, and also they release oxygen during this process, which increases the amount of oxygen in the air which we breathe. So the numeric equation for the process of photosynthesis is 6 moles of carbon dioxide react with 6 moles of water to form 1 mole of glucose and 6 mole of oxygen. So glucose is used to produce starch in plants. Can also convert glucose into cellulose and sucrose. So glucose is the main sugar that plant contain. Glucose is a polysaccharide, as you know. So many molecules of glucose join together to form disaccharide or polysaccharide. So as cellulose is a polysaccharide and fructose is a disaccharide. So the process takes place in the plant, but the glucose is the foundation of that process. So carbon dioxide, water, light, chlorophyll, these are all the suitable temp and a suitable temperature. These are all the requirements which are required for photosynthesis to be carried out. So as we know, life depends on photosynthesis. So what does this phrase mean? This phrase actually means that the start of every food chain is the producer, and the producer is the one that can convert light energy into chemical energy or can make its own food. So in this case, most of the time the producer is a plant. So plant, the life depends on plant, as it can make its own food, and the plant and other organisms feed on the plants. So the photosynthesis, the food chain starts from the plant. So as the food chain starts from the plant, this means that life depends on photosynthesis, as photosynthesis is a process by which plants create their own food. So the process begins and the food chain also starts from the plants. Second reason for this phrase is that during photosynthesis, oxygen is released in the environment. Oxygen is consumed by every living organism on this planet, be it birds, fishes, humans, any organism present in this planet uses oxygen to respire. So during photosynthesis, oxygen is produced, which is used by organisms in our surroundings. So there are some conditions for photosynthesis. Those conditions are carbon dioxide, water, light, chlorophyll, and a suitable temperature. So we'll investigate these conditions. How can we investigate these conditions? This is a very important topic. It is mostly assessed in paper 4, which is alternative to practical. So we can see the conditions needed for photosynthesis. So how can we investigate those conditions? So the first condition that can be there are only three investigations, planning of investigations in the syllabus, which is light, carbon dioxide, and chlorophyll. So how can we investigate for light? So first of all, we need to cover a leaf of a plant for a whole day. After covering the leaf of the plant, we'll set it in the sunlight for a whole day for the time the light is present. The covered leaf will be would be on the attached to the plant and would be set in the sunlight. After the sunlight has disappeared, we will test that leaf for starch through the process of iodine solution, as we learned earlier in the topic of animal nutrition or biological molecules. The test for starch is done by through iodine solution. So we will test for starch. If the starch is present, it means that photosynthesis has taken place. If the starch is not present, it means photosynthesis has not taken place, and the result for the test of starch would always be negative. As in the absence of light, no photosynthesis can take place, so the plants cannot produce their own food, which is starch or glucose. The next investigation would be of carbon dioxide. So we can place a plant inside a bell jar, and inside the bell jar we can place a dish filled with soda lime. Soda lime is a uh, you can say a substance which cancels out carbon dioxide. Soda lime would be present inside the bell jar, as it will provide a vacuum environment for the plant, so no air could enter the bell jar. So this means that no carbon dioxide will be present. So then we can again test for starch, and if the starch is present, it means photosynthesis took place, and if the starch is not present, so not photosynthesis took place. The next test would be of chlorophyll. So we can firstly de-starch. We can de-starch the leaf. We can put a de-starch variegated leaf plant in the sunlight, and then again test for starch. So the next test is the investigation for the rate of photosynthesis. So first of all, for this process, we can measure the rate of photosynthesis at the rate which reactants are being used up, or the rate at which the products are formed. So the rate at which carbon dioxide and water is being used up, or the rate at which glucose or oxygen is produced, is the rate of photosynthesis. So we can measure them. So for this, we need to cut a healthy piece of pondweed about 10 centimeter in length. Then we can place the specimen upside down in a boiling tube of water. It helps to sink the pondweed if you attach a small paper clip to the bottom of the specimen. Then we can clamp the tube to hold it upright in a beaker of water. The tube ensures that the pondweed is perpendicular to the source of light. The beaker of water is needed to maintain a constant temperature. Then we can place a thermometer in the water to record the temperature. Then we can turn off the room lights to reduce the background light and place a bench lamp close to the beaker. Then we can observe the specimen for several minutes. Then we should be able to see some gas escaping, which would be obviously oxygen, as oxygen is being produced. So the gas escaping would be in the form of bubbles. So we can count the number of bubbles produced for a whole minute, and we can repeat the process again and again 2 more times. So we'll have three readings, total amount of three readings for a minute. For example, the first reading is 28 bubbles per minute, the second is 30 bubbles per minute, and the third reading is 32 bubbles per minute. So we'll add this reading and divide it by 3 to take out the average per minute. So it would be a more specific answer. So this is the investigation of the rate of photosynthesis. Easy process. Then we can move on to the structure of the leaf. One last thing in this, the rate of photosynthesis increases as the light source moves closer. If the light source is closer to uh, your pondweed, the rate of photosynthesis would be higher, as more bubbles would be produced. So the bubbles produced, the production of bubbles should be at a moderate rate. So the lamp should be set at a specific distance. So it's easy to count the bubbles produced. Now next is the structure of the leaf. So as you can see here, this is the structure of the leaf. The upper layer, the yellow layer on the top of the plant, it is called the cuticle. The cuticle could be, you can say, the skin of the plant. It's a waxy material on the top of the plant. Then below that, the vertical green material, it's called the pellucid mesophyll, and the red circular material or the oval material, it's called the spongy mesophyll. The spaces empty between the spongy mesophyll is air sacs. This the opening in the plant are the stomata or the stomatal pore, through which diffusion takes place. The opening into the closing of stomata is controlled by the guard cell. You can also see phloem and xylem present here. So let's move on to the cuticle. So the cells of the upper and lower epidermis contains this layer of the plant, which is known as a cuticle. This cuticle is a waxy cuticle which reduces the water loss and acts as a barrier for the plants against disease-causing infections, such as insects or pathogens that cause diseases in the plant. The cuticle acts as a barrier and does not allow them to enter the plant, the leaf. It also reduces the water loss, so the plant does not uh, die. Next is the stomata or the guard cells. So stomata is the pore in the leaf, which is the stomata, and is surrounded by guard cells. Stomata allows the changing of air from the surroundings. So it is through the stomata the carbon dioxide enters the plant for photosynthesis, and through the stomata oxygen leaves the plant as a product of photosynthesis. Guard cells contain chloroplasts through which they control the opening and closing of the stomata. At night, as no light is present, the guard cells close the stomata, so no water loss takes place. Neither does the gas escape. The plant too need oxygen for respiration. So at night, the plants are net consumers of oxygen. In the daytime, they consume less oxygen than they're producing. So at night, the guard cells close the stomata, and at daytime, as the sunlight is present and the chloroplasts in the stomata detect sunlight to open the and the guard cells detect sunlight to open the stomata. So So next are the mesophyll cells. There are two types of mesophyll cells: the pelagoid mesophyll and the spongy mesophyll cells. Pelagoid mesophills contain many chloroplasts for photosynthesis to take place as it is closer to the light source. Spongy mesophyll cells have less chloroplasts but air spaces between them for CO2 and to enter and O2 to exit. So there are two types of spongy mesophyll cells. As we understand, the pelagoid mesophyll contains more chloroplasts as it is closer to the sunlight. So more chloroplasts would result in trapping more sunlight and producing more food for the plant. So more photosynthesis, so more food for the plant. And the spongy mesophyll, as it is further away from the sunlight, it contains less chloroplasts, but it has air spaces between them, through which the diffusion of air takes place, through which the carbon dioxide enters the spongy mesophyll and oxygen exits the spongy mesophyll. So the process of photosynthesis can be carried out easily. So the next is the xylem. So xylem is the, as we have studied xylem in very detail in the first chapter, which is the cells. So here we'll just have an overview. So xylem, the main function of xylem is to supply water and mineral ions to the leaf. Water is carried through xylem vessels. This movement is called the transpiration steam. Phloem. The products of photosynthesis, like sugars that are produced in the plants, are transported away from the leaf to the other parts of the plant where photosynthesis does not take place. For example, the movement of sugars through the phloem is called translocation. For example, the roots do not have chloroplasts, so this means that roots cannot carry photosynthesis. There's no light reaches the roots, so roots cannot make their own food. So the glucose or the starch made inside the leaf is transported to the roots to grow through the phloem. Xylem and phloem are collectively known as vascular bundles. So the topic of plant nutrition today has ended. We'll meet in the next video, which would be on transport in humans.